So I'm going to talk about a project that's called the Million Person Project at the healthcare system I work for, Providence, that has the ability to catalyze a real paradigm shift in healthcare. And you'll come to understand all of this as we go on. So I'll start by giving you a background and telling you what 21st century medicine is, in my view. So I went to Caltech as an assistant professor in 1970, and I was really impressed with the complexity of human beings. And this led me to realize if I ever wanted to understand either wellness or disease, and I had an interest then in both in a general way, that I would have to introduce both tools and strategies that allowed us to deal with human complexity. And in the course of that, I did either started or coordinated with 7 different paradigm changes in modern biology, and each of these did solve in varying ways aspects of human complexity, but they also framed my view of 21st century medicine. So let's see if I can get rid of this. So the first paradigm change then was to bring engineering to biology. And for a period from 1970 to 1992 or so, my laboratory developed 6 different instruments for reading and that is sequencing proteins and genes and synthesizing proteins and genes. And these instruments served as the framework of modern technology for biology. They also allowed us to do high throughput measurements on human beings. And they also started the accumulation of data that led to big data and its analytics, which you've all heard about. One of these instruments, the automated DNA sequencer, got me invited to the first meeting ever held on the Human Genome Project in spring of 1985, where 12 of us came together to pass judgment on whether it was a good idea to do the Human Genome Project or not. And we came to 2 conclusions. One, that it was technically feasible, although at that time really difficult. And 2, we were split 6-6 on whether it was a good idea. And those against it were terrified that the big science that Genome Project represented would swallow up the smaller, more traditional small science that most biologists at that time practiced with. Generally, labs had had a senior person and say 3 to 10 technicians, graduate students, postdocs, and the like. But it turned out that fear was really unfounded. And what the Genome Project did, one of the things it did from my point of view, was it gave us access to human genetic variability and the ability to correlate it with wellness phenotypes or disease phenotypes. And that's an important point we'll return to later. In developing the automated DNA sequencer, I had to recruit together a molecular biologist, myself, an engineer, a chemist, and a computer scientist to put together the technologies that was necessary to automate sequencing. And that made me realize that if we created a department of biology at Caltech that was cross-disciplinary in nature, it could really fuel a lot of technology development. And biology turned out not to be interested in that. So Bill Gates made it possible to go to the University of Washington in 1992, and I started there, the first cross-disciplinary department. And it was spectacularly successful in the 8 years of its existence. We developed key techniques for genomics, for proteomics, for cell biology, for single cell analysis, and similar kinds of things. But when I moved to the University of Washington, I really wanted to build on this cross-disciplinary platform, a systems biology center or institute. And it turned out it was just too difficult to do it at the University of Washington, largely because of bureaucracy, frankly. So in 2000, I actually resigned from the University of Washington and started the Institute for Systems Biology in Seattle that pioneered systems biology. We developed a global, holistic approach to doing biology and taking on problems of disease. But I think in many ways, one of the most interesting things we did in the early 2000s at the Institute was to really formulate the concepts that stood as the framework for 21st century medicine. And at the heart of it, it was the conviction that medicine should be predictive, preventive, personalized, and participatory, and that this so-called P4 medicine or healthcare really had two central domains, a wellness domain and a disease domain. And of course, at that time in the early 2000s, most health systems didn't even think about wellness at all. We developed some additional technology, and then in 2014, a colleague of mine, Nathan Price, and I persuaded 108 of our friends to participate in a pilot program to study wellness in a quantitative fashion by doing complete genome sequencing and longitudinal phenome analyses that I'll define in a couple of slides. And that program was so successful in one, proving that quantitative or scientific wellness really can improve the health for an individual. And two, it demonstrated the power of the longitudinal data clouds to decipher mysteries of both biology and disease. And we'll talk about that in just a moment. And the last paradigm change came when the CEO of Providence came to me in 2016 and asked if I would like to be chief science officer for them and to affiliate ISB with Providence. And because I saw this as a unique opportunity to bring 21st century medicine, which I'll define in just a moment, to a healthcare system, I accepted the offer and immediately formulated a new project called the Million Person Project, which again, we'll talk about in just a few moments. So 21st century healthcare is about the idea that medicine must be practiced at the level of each individual. We call this an N of one medicine. And the idea is that you follow the health trajectory of each individual and you optimize it. And if you think about it, most individuals' health trajectory is made up of three different components. There's a long period of wellness, and we hope to extend that period significantly in the future. And then most people actually transition from wellness to a disease. And at that initiation point, the disease is obviously simplest. And the idea that we hope to be able to practice in the future is that we can identify that transition and we can develop a therapy that will reverse it before you ever see any clinical signs of the disease. And this is called prediction and prevention. It's the first two P's in the P4. And of course, the third stage is when you have a disease, then follow its progression up to the point that you get clinical signs. And when you get clinical signs already, the nature of the disease and its disease-perturbed biological networks is very complicated. And for many chronic diseases, including Alzheimer's and obesity, a single drug is never going to work to cure it. And in fact, all of contemporary medicine now operates at the level of studying disease after the clinical signs have come. And we want to study disease at their various early detectable stage. So 21st century is all about emphasizing wellness and optimizing healthy aging. It's all about identifying wellness to disease transitions and reversing them. And it's all about dealing with disease far more effectively than we do now. And we'll talk about Alzheimer's as an example of how we might do that in just a moment. If you think about contemporary medicine, these are four qualities that we have to be concerned about. One, the quality of the US medicine. We're 19th or 20th out of 19 developed countries, even though we spend probably 5 times as much money per individual on healthcare as the next closest developed nation. We're really worried about the aging population, which is expanding very, very quickly. And we're seeing an explosion of chronic diseases, particularly Alzheimer's, diabetes, metabolic disease, and the like. And finally, the cost is just prohibitive, and we have to turn that around. And the argument that I'll make is 21st century medicine deals with all four of these challenges really very, very nicely. So the kind of analysis we did in the pilot project and for scientific wellness in Aravale, bringing it to consumers, was to determine the whole sequence of an individual's genome, and then every 6 months to do blood tests that analyzed about 1,200 analytes, that is, clinical chemistries and metabolites and proteins. And we, every 6 months, analyzed the gut microbiome for the ratio of microbial species. And then finally, we used digital monitoring with a Fitbit to track physiology and lifestyle in various ways. And this generated for each individual a unique data cloud that, when analyzed, led to a series of actionable possibilities that, if acted upon, could lead to improved wellness and or let you avoid disease. And as the data increases, the number of actionable possibilities is actually increasing exponentially. So this did two different things. One, it validated the power of scientific wellness. And two, it gave us data clouds in Aravale for 5,000 people that generated fantastic new insights into biology and disease. And I'm only going to talk about 4 of these insights, but they're very central things we can do. So we were able, as I said, to validate by appropriate measures the effectiveness of scientific wellness. And we were able to actually improve strikingly the quality of life for many ordinary people that didn't really have diseases. A second thing we were able to do from the 5,000 individual data clouds was to bin them into 10-year categories, 20 to 30, 30 to 40, up to 80 to 90, and to show the level of analyte expression and their control of expression decreased exponentially as one aged. And without going into any detail, we were able to calculate for each individual a biological age, that is, the age your body says you are. And if your biological age is lower than your chronologic age, you're aging in a healthy way. If it's higher, you're not aging in a healthy way. We demonstrated, for example, for the 5,000 people in Airville, on average, the males lost 1 year per year in biological age, and the females lost 1.5 years per year of biological age. And so this metric validated our ability to follow wellness and to assess whether it's going in the right direction. We looked at 40 different diseases, and for individuals with those diseases, in each case, they had a biological age well above their chronologic age. So you can actually use biological age in another way, because the blood analytes that we use to determine the biological age give us clues for each individual as how we uniquely can optimize biological aging. So we put together then scientific wellness with biological aging and its optimization, and we call this healthy aging. We were also able to take the genetics of individuals, more specifically genome-wide association scores, and we could actually, because we had the whole genome, go into each individual and determine for 54 different diseases what their genetic risk was. And for each of the diseases, we bin the 5,000 people into these 5 categories: very low risk, very high risk, and 3 in between risks. And what we demonstrated that was really interesting is, for example, if you had a very high risk for LDL cholesterol, which is a proxy for heart disease, the only way you could bring down that LDL cholesterol is with statins or other drugs. Whereas if you were at low risk for LDL cholesterol and had high cholesterol, you could bring it down beautifully with diet and exercise and the like. And the importance of this for the 54 diseases is we'll end up treating people at low risk for a disease differently from those that are at high risk for that disease. And the final really interesting observation was that we were able in these 5,000 people to identify 167 wellness to disease transitions. And they transitioned to all of the major kinds of chronic diseases. In fact, 35 of these transitions were cancer. And the illustration that I have here was the first one we studied, a 57-year-old woman that was diagnosed with stage 4 pancreatic cancer. And we had 4 blood draws prior to her clinical diagnosis, 6 months apart. And we analyzed each of those blood draws with regard to the 1,200 blood analytes, asking were any of them outliers when we compared them against the normal distribution of about 2,000 individuals we had at the time this woman was diagnosed. And we found a whole series of proteins that were outliers. And what this means is we can detect the transition to disease years before the clinical diagnosis comes. And in fact, we could detect it up to for the whole duration of when we had blood loss, which was about 2 years. And our estimate probably is the real initial transition happened even 2 or 3 years earlier. So if we had longitudinal data for a 5-year period, we'd be able to see the earliest transitions. And the idea is we can use that outlier as a biomarker to locate the point of transition and then take systems approaches to identify therapies that will reverse the disease before it ever manifests itself as a disease phenotype. And in fact, what's interesting about these things is, on the one hand, biological age leads to optimizing and creating a healthy aging profile for you. And the transitions in time will let us eliminate chronic disease by removing them before they ever manifest themselves as a disease. This is prediction and prevention, and it's the essence, and a very important essence, of 21st century medicine. So let me talk about studies we've done on Alzheimer's and show you how we can use these ideas in 21st century medicine to be really effective. So we can do the genome-phenome analysis of each individual. And the idea is, what we really want to do is use another tool, which is called PET scanning, metabolic PET scanning, glucose PET scanning, because that has the ability to detect Alzheimer's 4 to 10 years before any clinical diagnosis is made. So we're using this deep phenotyping to follow high-risk Alzheimer's patients that are still normal, to see when they first transition and get a biomarker to mark that transition point, so that we can begin to develop a therapy that will reverse the disease very early before it ever manifests itself. And what kind of therapy will we use for Alzheimer's? Well, this is really interesting. Alzheimer's is a disease that has had 400 clinical trials in the last 10 years. Not a single one worked. And it turns out that an individual named Dale Bredesen pioneered a whole new approach to Alzheimer's by asking the simple question, how can I optimize synaptic communication? That is, how nerve cells talk to one another. Because this is one of the things that gets lost in Alzheimer's. And he came up with a 36-point multimodal therapy to do that. He did an observational trial on 100 individuals with very early Alzheimer's and showed that more than 85% of them with this multimodal therapy could be returned to normal. There is a second multimodal therapy that was pioneered by Michael Merzenich, who was at UCSF, who essentially demonstrated that the brain has the ability as we age to lose 25 or so cognitive functions. But what you can do with computer games is design them so they'll return lost cognitive functions. And examples are reaction time, the memory, the ability to see a wide peripheral field, and so forth. And he's demonstrated in a whole series of clinical trials that individuals at high risk for neurodegenerative disease really can prevent that by using these games, which are called Brain HQ. So we've started at ISB now 3 different clinical trials using these multimodal therapies on early stage Alzheimer's patients. And the early results really look extremely promising. So the idea, we hope in the future, is that we'll have a biomarker for the earliest stage of Alzheimer's, and we'll immediately be able to reverse quite easily that individual back to a normal. And that's what we hope to be able to do for many different chronic diseases. So what exactly is the Million-Person Genome Phenome Project? Well, it's recruiting 1 million Providence patients together with their physicians. And that's a big challenge, I will admit, to educate and recruit and get the appropriate permissions and so forth. And then from each of individual drawing blood and getting stool samples for a biobank. And the biobank then will send these samples out to companies that either do genome sequencing or the various longitudinal phenotyping measurements of the blood analytes and the gut microbiome. And the idea then is once those data are returned, we can put them in the cloud and bring with each patient their electronic health records, and then we can analyze them using machine learning, AI, artificial intelligence, and actually knowledge graphs, which is a very powerful new approach that's being developed. And once we've done that analysis, it will lead to actionable possibilities that will improve patient health or help with partners' desires to be able to create products for the marketplace. And we have new approaches to clinical trials that I'll talk a little bit more about in a few moments, and interesting new, in many cases, digital diagnostic products. Now, to do this ambitious project, which is very costly, we're in the process now of recruiting potential partners, and some that I've talked to are listed here. And we have a fair amount of interest, and the partners will do two things. One, they'll help us analyze the data in the context of their own interests. And two, they'll help pay for generating the data that lets us create this enormous database of things. And what's interesting is we started at the beginning of this year a pilot project for this project with 5,000 individuals to basically learn how to do all the things that I've listed right here. The knowledge graph I mentioned about is really nothing more than a database that combines together all biomedical databases so you can have a one-stop shop for all medical knowledge. Now you have to go to a whole series of different databases for proteins or for metabolites or for transcription analyses, all of those things. And in the future, it'll all be in one database that you can access all of these things in exactly the same kind of way. So we can use these knowledge bases to connect the dots that puzzle us in new ways. And the really spectacular thing we've already demonstrated works is you can put the data of a patient into it, and you can get very fast, specific diagnoses of just what that patient has. That is, it will facilitate N-of-1 medicine and really let us know what's going on. So what we plan to do is have partners that fall into these different concentric circles, the inner partners more heavily committed to the program than successively outer layers of partners. And inner partners would have more control over regulation of the program and so forth. And likewise, by 2025, we will have enrolled 1 million research participants in this precision population health program that really will create totally unique assets. So the idea that open innovation will allow our company partners to work individually together with other companies or together with ISB. The insights will come from machine learning, AI, knowledge graphs, and so forth. We've hired talented people, both at the research, clinical research level and the data science level, that are already initiating this project. We're beginning to educate physicians and participants and create the materials for doing so. We're creating this big data commons that people will use, and we'll have an engaged population of patients that can be used for very quickly for a wide variety of clinical trials if they consent to do so. The biobank will be enormously valuable because it will have thousands of samples that we can return to query biological hypotheses and see what the underlying mechanistic possibilities are for given disease, given transitions, and things like that. And we're going to be able to create health data platform that I think is absolutely going to revolutionize medicine in the future. And the kind of data that we'll use is the biological data I've talked mostly about, or the social determinants of health data that takes into account the things your environment brings into you and your personal behavior brings to you. And then we can do claims data. Was it successful or not? And then we can look at clinical trial data in the electronic medical records. And these will transform how we learn about wellness and disease, how we organize and deliver care, how we measure, price, and manage risk. And finally, it'll let us innovate and develop all sorts of new kinds of products. So there are 8 other groups that are proposing large genome-like projects, but none of them are doing longitudinal phenome. That is the unique feature of the million-person project. The other thing that is unique is the million-person project is the only one that's driven by this vision of 21st century medicine, where we optimize it, deal with it as they go through 3 phases at various portions of their life. And on the left are a whole series of features that are desirable. And again, the million-person project is the only project that actually does all of those things. So it is a really unique and I think a transformational effort. Now, Providence is a terrific partner for this project because it's a very large healthcare, the 3rd largest healthcare system. It spans the entire coast, and that covers many different racial populations, obviously, as well as Montana, New Mexico, and the western part of Texas. And it's a system that has 51 hospitals and roughly 25,000 physicians, and of the order of 14 million patients with their electronic health records and so forth. So it is a healthcare system that will be able first to scale to the million-person project and then scale to multi-millions of patients being engaged in this kind of program. And I think ISB is a great leader for the million-person project. We're small, 12 faculty, 180 staff. We've pioneered systems biology. We've started 19 companies over the 20 years, and we've successfully executed very large science projects. And we have a good blend of biology, technology, computational skills, and medical expertise. ISB is interactive. We can make quick decisions, small chain of command. And it's really pushing the million-person project and something I call deep immunologic phenotyping, which we'll talk about in a moment in the context of COVID. We have a connection through me that's between ISB and Providence that is very intimate. And Jim Heath's president is a wonderful diagnostic expert, a chemistry, broad background in chemistry, and is now an expert in immuno-oncology, a major part of cancer biology these days. We have a major effort on these knowledge graphs I've talked about, and we're really interested in scientific wellness and healthy aging and how we optimize these in individuals. And we have 6 full-time people working on education, and we operate at the K-12 level, junior college, graduate student level, and physicians level. And, I, for example, have written 6 textbooks that span immunology, molecular biology, biochemistry, genetics, and most recently, systems biology. So we're a thought leader in these multi-omic analyses, and over the last 10 years or so, we've published a series of the papers that have really led the field in the evolution of the population field for longitudinal multi-omic analyses of individuals. And we've set up now 11 clinical trials, of which I've only listed 5 here to give you examples. But we did a 2-year clinical trial with Airavail on scientific wellness that really validated the power of scientific wellness to improve the health of all the individuals who took the program seriously. And most recently, we've started a COVID-19 clinical trial, which I'll talk about in one slide. So as I said, we started the pilot program in January of 2020 to get 5,000 patients in the pilot, and were interrupted by COVID-19 in pandemic in February of 2020. And it's now delayed either to the winter of 2020, or more likely it's going to be early spring of 2021. But what we did when the COVID hit in Seattle was one of the very first locations it hit because of our proximity to China, was create an observational study, and I'll define what that means in just a moment, that used the genome-phenome analysis that I've talked about and something new, which is deep immune phenotyping, and I'll describe that in just a moment. But what's been interesting about COVID is it has been a grand disruptor of healthcare, but in a curious way, it's really enabled the million-person project to move forward in a very effective way. So the clinical trial we did was an observational trial. And what that means is you take patients as they arrive at the hospital and you follow them, and you follow whatever's being done to them. If they come in on drugs or if they're given drugs, you don't manipulate them at all. You just observe what happens with each individual patient, because the deep phenotyping lets us derive enormous amounts of information from just a single patient. So we're obtaining 300 patients, and we do blood draws at diagnosis 10 days, and then if and when they go home, at convalescence of about 40 days. And it includes a lot of Hispanics and African Americans, as well as Caucasians and Asians. The assays that we're doing are the genome-phenome assay I talked about earlier, but the deep immunologic phenotyping takes 5,000 white blood cells at each blood draw for each patient and does single-cell analysis. The whole transcriptome, that is the whole population of messenger RNAs, we measure 200 cell surface proteins, 40 secreted cytokines, and their T and or B cell receptors. And we identify the HLA locus, because that's the locus that is used to present antigen to killer T cells from the epitopes on the virus. And so we've been able to study all of these different aspects in enormous detail. And this has allowed us to align all the patients because we have fingerprints for the progression of the disease. We have fingerprints for the trajectory of the two major types of drugs that are being used on these patients. And it's been absolutely revelatory. And in fact, we've now deeply analyzed 129 patients and have a cell paper in press on this deep immune phenotyping. Okay, so what has the COVID study done for us? One, this observational study was started within 10 days of the time we proposed it to Swedish Hospital, which is a record for getting things done. And the emergency imperativeness really facilitated this. Number two, we were able to go to top immune researchers across a spectrum of institutions, Stanford, UCSF, the Fred Hutch, Rockefeller, UCLA, etc., to be able to gather the expertise we needed to be able to translate the enormous deep immune phenotyping into results. We were able to recruit of the order of 10 small companies that had unique abilities for data generation to help us with the generation of unique aspects of data. And finally, when I went to number 2 in Merck and told him about the deep phenotyping program, within 20 minutes, he agreed to fund the entire program and to do it where the data would be open and to do it with other pharma. And these were all paradigm shifts in how pharma usually treats academic projects. And the money we got from the clinical trials, this allowed us to build a computer and clinical scientist team that's really uniquely powerful. And we've created a series of platforms to do all the complicated things I described on the project for the million team. So what are some of the results that we have? We've got 5 patients that were infected twice. And the first one, which we've studied the most carefully, the initial infection was the Chinese virus. The second infection was the European virus. They differed by 8 mutations in the viral genome. And mutations at key points that made the European virus significantly more infectious. But what was interesting is we took antibodies from the first infection, the Chinese virus infection, and showed they didn't work well at all in the second infection. And that clearly has vaccine implications. A second thing that we did was to use the CD8 T cells and the antigen presenting machinery to identify the viral epitopes Caucasians and Asians used. And it turned out that they used quite different epitopes. And again, that has interesting applications for vaccines. We were able to follow the shift in CD8 killer phenotypes either as the disease progressed or as it resolved, and gave us deep insights into predicting the course of disease in the individual. We defined 3 new types of cells that we're now still looking into to really understand what they do. And maybe the most startling observation was that we were able to demonstrate that in people that came in with the mild disease, that is stage 2, 3, where the WHO goes up to 8, that patients at that stage demonstrate almost a complete loss of essential amino acids fatty acids, phospholipids, many other metabolites. And it is as if the virus is signaled, build me a lot of immune cells rapidly, and the body goes out and scarfs up all of the essential building blocks for cells. And we're testing this hypothesis in interesting ways now. And again, the deep immunologic phenotyping allowed us to assess trajectories for drug responses for these two drugs. The first, an anti-cytokine killer, a blocker, IL-6, and the second, an antiviral, to show that the trajectories were distinct. And they gave us clues as to how the drugs acted and how you should design clinical trials to optimize the features of each drug. What is interesting about these studies is we're beginning to realize that being able to generate this much information on individuals means that the deductions we can draw from even a single patient are powerful. And we went on first to phenotype 30. We learned a lot of new things. By the time we got to 100, now that we've done 150, we saw that at 100, we'd learned most of the major things we were going to learn. So what we were able to do is go into the Providence database of electronic medical records and identify 1,500 patients with COVID. And we're mapping what we've learned in our smaller number of patients into these electronic medical records to get biomarkers that are common, used in ordinary medicine, that will be proxies for the interesting transitions we've seen with the patients. So we think in the future we'll be able to design optimal clinical trials with 100 individuals. And if we do so successfully, it can lead to FDA approval and so forth. And I'll just say that deep phenotyping we did here was immune, but we can do deep phenotyping metabolic, hormonal, neurologic, or other kinds of things, and will really transform our understanding of the biology and the disease that are related to those different kinds of organ systems. There are really important business opportunities for the Million Person Project. And here I've listed 21 different possibilities that can affect the care that's delivered to patients, the first 10, or can create intellectual property for business partners that will be profitable, the next 9 or so, or it will change how we carry out the research enterprise. And I'm not going to go into it, but there are a whole series of ways clinical trials can be done now that really are going to transform how we think about doing this in the future. And in fact, some of the more interesting examples of short-term opportunities, and this is the goose that laid the golden egg, is for 21st century CROs, that is 21st century clinical trials, we'll be able to operate them in a distributed manner where we can control computation, we can interrogate computationally the population that can be in 5 different states and identify the individuals that could be in a particular kind of clinical trial. We're going to actually move this scientific wellness from hospital patients out to consumers, and we think we can drive a consumer data-driven scientific wellness and healthy aging that will be transformational for people on the outside of the healthcare system. We're also interested in clinical studies with these deep omic analyses for all of the chronic diseases, where we'll look at those early transitions and come to understand in enormous detail what are the disease-perturbed networks that we can design simple therapies to reverse at the earliest stage. We're also interested in creating biomarkers for the stratification of disease into its different subtypes and the patients into different types of responses to the disease, and companion diagnostics, which will tell us which individuals will respond to a particular kind of drug. We're also thinking about two-step clinical trials of 100 patients each. The first to look at the drug and determine a biomarker panel for those that respond, and then in the second to use 100 responders to get a 96% success rate, and again, FDA approval for that kind of thing. We're going to be using AI-based recommendations because with the Million Person Genome Project, we'll literally get thousands of additional new clinical actionable insights, and AI will be necessary for each individual to identify actionable possibilities relevant to them and prioritize them, and then to educate the physician with regard to what these things mean. And that's going to be a fascinating challenge. And finally, we've demonstrated in a number of ways I haven't talked about that these data give us statistical approaches to biomarker and drug target discovery, I think that can really revolutionize how pharma actually operates. And the last example I'll give you that's something very interesting is we're developing a microfluidic device that can be used at home and self-administered either to detect COVID from saliva or to determine whether you have flu or COVID, which we'll need to be able to do this winter. And we will be able to get results in under an hour. It uses a powerful new amplification technology, which does not require temperature cycling, so you can make a very simple device. And it's going to be incredibly inexpensive. It'll be a two-part device, the major framework and the inner thing, which is you use for each test. And the cost of developing the inner device are pennies plus about $1, and the reusable module is $2. So for $3, you'll be able to get at home a COVID test. You'll be able to do it with your saliva, and you can do it as frequently as you wish. And the typical tests now are north of $125 per test. So it's strikingly different. So what should you take home from this? Well, I think the thing to remember is 21st century healthcare is N of one medicine. It's all about following the health trajectory and optimizing it across these three states: wellness, transition, wellness to disease, and finally disease. A second point is the importance of the individual in medicine. In the past, all medicine was done by averaging things, and it's turned out to be really ineffective in 50 different ways. And there's a book by a professor at Harvard called The End of Average, and he showed exactly the same is true for education, industry, and consumerism. It's one of the most interesting books I've read in the last 5 or 6 years, and I highly recommend it because although he doesn't do more than just barely mention it, it's really true of healthcare too. Healthcare is going to be about individual care. So some innovations of the Million Person Project, we're going to have many, many new clinical possibilities. We're going to have many new clinical research possibilities, new types, different types of clinical trial and so forth. And finally, there'll be the ability to spin off companies and collaborate with existing companies on opportunities that I think will transform medicine itself And finally, I would argue that 21st century medicine will leadership for the early adopters. And so, an interesting question is your own institution and where it stands with regard to this. And my final point is if we go back to the four uh, contemporary challenges of medicine, quality, aging, explosion of chronic disease, and cost, you see that for each one, we have powerful avenues that we can employ to improve quality, to deal with the aging population and the explosion of chronic diseases. And finally, all of these things will contribute in striking ways to reducing the cost. And just a simple example, suppose you could do cancer clinical trials with 200 people rather than with 20,000 people. Imagine the cost savings on that. So, 21st century is medicine, and P4 medicine will provide many additional approaches to each of these challenges. So, with that, let me just give you close with advice to students. So, you shouldn't do science unless you think it's fun and exciting. For me, I can't even imagine another career, and it's kept me fun and excited my entire lifetime. Work with outstanding colleagues and mentors because they have the ability to stimulate and push you in directions where ordinary people aren't ever going to do. You need to become cross-disciplinary scientists to have domain expertise in biology or medicine, whatever your area is. But you'll need computational skills and statistics because big data is here to stay and it's going to be an intimate part of anything you're going to do in the future. Working on big and challenging problems should go without saying. You don't want to be a librarian just cataloging things that are already known. You want to move the frontiers forward. That's where you get enormous excitement. And I think maybe the most important advice I could give you is really think about changing directions every 10 to 15 years, as I did when I went through the paradigm changes I talked about earlier. Because what happens is every time you change direction, a career, take a new career trajectory, you become insecure, you learn a lot, you meet new people, and you think about things in new and different ways. And it's absolutely a rejuvenation of what goes on. I had a columnist recently said, well, what are the most important changes in your career for you? Or what are the most important things that have impacted your career? And I said without hesitation, it's every move I've made. When I went from NIH to Caltech, that opened up my thinking, totally put me at a different spectrum. When I went from Caltech to the University of Washington, I moved into medicine, learned a lot, started over again in many ways. And when I started systems biology, exactly the same thing happened. And becoming chief science officer at Providence has been an incredible yet another learning experience. So don't do what you've done for your PhD thesis your whole life. Change directions and retain your vitality and your passions and so forth. So that's it. I'm finished. So I'll stop sharing now. And I don't know what you want to do. I'd be glad to answer questions or whatever you'd like.
Awesome. Thank you so much, Dr. Hood, for such an informative talk. It was so inspiring to hear about like all your accomplishments and your progress for your research. I really enjoyed it. And personally, I'm from Seattle, so it was really cool to hear about the COVID immune phenotyping research that's been happening like right here at Swedish Hospital. That was
Cool.
really cool to hear about. So now I'm gonna pass it off to Natalia.
Oh yeah, I just wanted to say how fascinating it is about the many significant opportunities that the Million Person Project may bring. And thank you so much for sharing that with us. And I wanted to ask everyone, like you said, if anyone has any questions, please feel free to type in the chat, or you can ask us to unmute you. I see three questions in the chat already, Dr. Hood. Would you like me to read them out loud for you,
Yeah,
or?
I don't have chat, so you'll have. Oh,
Oh,
I
okay.
do have chat over here. Uh, well, why don't you ask them? That's easy enough.
Okay, so the first one says, did you know that wellness was something that you wanted to explore in your research when you began, or did it come about after a series of other research projects?
I would say at Caltech, I knew when I started as an assistant professor, I knew I wanted to understand health and disease. I don't think I'd really articulated wellness until really going to the University of Washington and then starting to see the contrast between wellness and disease. And you know, the fact that a really good medical school was focused entirely on disease and not at all on wellness. And that's why we started this series of really seminal experiments in 2014 to begin defining wellness in a quantitative sense.
That's so interesting. Thank you. So the second question is from Sonia, and it says, is risk level for various diseases something that you use machines or programs to determine? And if so, what kind of machines or programs? And there's a second part to it. So instead of doing that, did you and your team manually have to go through the data collected for each of the 5,000 individuals and determine risk level for each disease that way?
So the answer is you there were a series of population studies with up to 500,000 individuals that looked at genetic variants that controlled uh, cardiovascular disease, obesity, Alzheimer's, and things like that. And what they found in these analyses generally were a whole series of variants that made very, very small contributions to the disease. Okay. But they identified them. And because we'd done the complete genome sequence, we could actually map the level that variant contributed to the disease into an individual's genetic risk. So the genetic risk was basically a genetic calculation that came from having the genome sequence data that we could compare against the genetics and so forth. What we've done is develop computational techniques that enormously automate the ability to map these disease risks into an individual's genome. So we did a study that was published in NAS just a couple of months ago where we looked at 54 different diseases and we assessed the genetic risk for these individuals and made some of the conclusions that I talked about in my talk. But with big data, the key thing is you have to have platforms that automate not only the acquisition of data, but its analysis in a whole series of different kinds of ways, because big data is well beyond uh, an individual looking at things one at a time. It's kind of like systems biology. We don't look at biological systems anymore, one gene or one protein at a time. We almost always think about at least biological networks, which are a subset of the system that goes to create a particular phenotype, wellness, and or disease.
It's crazy how many different parts there are to one single experiment or project.
Yeah.
So yeah, the next question is again from Xavier, and she asked, were there any environmental factors you noticed either that were either positively or negatively affected a participant's progress in the Alzheimer's disease study?
So the answer is yes. An absolutely major factor for Alzheimer's are yeast or mold infections that can get into your body. And I mean, there are really dramatic cases where we're able to measure these various types of fungi and so forth. And there are really dramatic effects where if you just move the person out of the house that has black mold, within 6 months, their signs start to reverse as the black mold is expelled from the system. Or you can use more active techniques for chelation and removal of mold from individuals, and you'll get them cured up more easily. But in scientific wellness and Alzheimer's in particular, there are a lot of toxins that you have to be very careful about. Many different types of poisonous species. But the worst one to watch out for is mold that comes from roofs that leak, because this is a really common place where black mold sets in, and black mold is very bad for human beings. And if you live in a house that has black mold spores all through the air, you're just asking to transition yourself into an early stage of Alzheimer's disease.
Oh, that's so interesting. I had never heard that before. So we
I'll tell you also, in scientific wellness, we were able to show in the first study of 100 individuals, we were able to show 3 people that had very high levels of mercury. In 2 cases, the mercury came from eating excess tuna sushi. Tuna is a fish that's at the top of the chain, and it's accumulated all the poisons that all the smaller creatures that have been eaten by the next size creatures have. So tuna is a very bad place if for mercury. And what happened is the 2 people that had those high mercury levels just stopped eating sushi, and the levels came down in a period of 6 months to normal. And the other person had high mercury because the old amalgam fillings of individuals were very high in mercury. And she just went in and had her old fillings replaced with new ones, and that changed her mercury levels in an appropriate period of time again. So the more environmental toxins you can measure, the better off you are. The trouble is, everything you measure costs money, and you kind of have to optimize how much money you have to spend on people to optimize their wellness.
Oh yeah, I eat a lot of tuna. I have to change that. Xavier
Well, just
also
get
said
your
the
blood
same.
mercury level. There are people that have variants that can clear tuna beautifully. And if
Oh,
you have that variant, you can eat as much tuna sushi as you want.
oh, I hope I have that variant then. So we have 2 more questions. Is that okay? Do you have enough
Sure.
time to answer them? Okay.
Sure.
So one is from Lena, and she asked, do people respond positively to knowing about their genetic risks? And how often do they end up listening to the preventative methods and implementing them in their own lives? Is this information too depressing for patients with high genetic risks?
Well, the important thing that I didn't mention in Airvale and in the 100-person Pioneer Project was we had wellness coaches that were trained in psychology so they could persuade people to change their behavior. And they were trained usually in diet or biochemistry or something like this. But the point is, those wellness coaches became your mother. They delivered the actionable possibilities to you. They explained them in detail. And they were incredibly successful in persuading people to change their behavior. Now, the problem with that model is, let's say we go from 5,000 to 340 million people in the US, okay? Think of how many millions of coaches you'd need to do that. It doesn't scale. So we're actually talking with Amazon right now about the possibility of health avatars, where each person can have their own personal avatar that will receive the data and explain everything to them and encourage them and give them all the arguments as to why you want to do this to improve your health and so forth. So the answer is, with regard to genetic risk, if you just go up and tell a person that, hey, you're at high risk for ALS, they don't want to hear it. That's the last thing they want to hear. And there are two facts about that. One, education about what genetic risk can do for you is really important. And two, we'll only tell patients about things where we can improve their health. If there's something that you can't do with regard to genetic risk, your doctor will know about it. So in the future, when a therapy comes, you can be exposed to it. But we'll only talk to people about things we can do something about.
Thank you, Dr. Hood. So the last question is from Naivat, and he's asking, the fungal infection that you spoke about, are there instances where individuals from the study who have lived in a particular environment with black mold for so long, when the effects are unreversible?
So I think the answer to that is yes, because once you move beyond the early diagnosable stage of disease, you begin to lose neurons. And neurons are not easy to reproduce and refurnish. So depending on the stage of Alzheimer's disease that you're at, it may not be reversible. And what we know now is, if it's for the earliest clinical stage, we do know we can reverse that. But you go through a 5 to 10 year continuum where you gradually get worse and worse and worse. And we know at the end stages, there's nothing that can reverse that. So that's why early detection and reversal is key. You don't ever want to transition and progress into disease for a long period of time. And what's true of Alzheimer's is true of all of the other chronic disease. Diabetes, it gets to be absolutely reversible at probably an earlier stage than Alzheimer's does. So all of these things, you want to do something about them right away.
So in regards to this trajectory, I do have a question. Is it okay if I ask you?
Sure.
Okay. So I learned this week in one of my classes that there are two proteins involved in the Alzheimer's disease, which are beta amyloid and tau proteins. So I was wondering if there's any way to detect their abnormalities.
Just a sec. Someone's knocking on my door. I'll be right back. Okay.
No problem.
Oh, okay. Hi, come on in. I'm on a seminar right now, so Becky will be down.
I'm sorry.
Yeah, I mean, the two are tau and beta amyloid, and those are the proteins that was the focus of these 12 years of clinical trials that failed. And I think what is really clear now is those proteins are associated with Alzheimer's, but they are a cause of it rather than triggering the Alzheimer's itself.
Yeah, so I was wondering because they end up accumulating inside the neuron and also producing those tangles, which leads to the shrinkage in some brain
Sure,
areas.
sure,
So
sure.
I was wondering if there's any way to have an early detection in their abnormalities.
Well, I'll tell you, there are people that have bad Alzheimer's that have neither of those proteins. Okay. There are people that have a lot of those proteins that don't have Alzheimer's. So, what I'm saying is, it's not easy. It's true. The accumulation can destroy nerve cells and so on. If you're thinking about causes and early changes and stuff, you have to go elsewhere. And I think this multimodal therapy I described earlier is really where you have to go. I mean, some of the best drugs, there were really great drugs that could remove those proteins from the brain, and it didn't make.
So much for sharing that with us.
Sure, sure.
So, thank you for coming to our event today. I don't think that we have any other questions. And we really appreciate you taking the time to speak with all of us. And we're sincerely
Okay.
inspired by all of your work. Right now, we wanted to introduce the two clubs or organizations that are here today to everyone who's attending today's event. So, Dr. Hood, would you like to stay and listen to the introductions, or if you're unable to, we will understand.
You know, I can't because I have another Zoom call in 10 minutes.
Oh,
So
okay.
I'll let you go ahead and enjoy yourselves. And I enjoyed your questions and good luck on your careers. I think it's an exciting time for science, and I hope some of you choose to take that career path.
Thank you so much,
Okay,
Dr. Hood. I really appreciate it.
bye bye.
Thank you so much. Bye-bye.
Yep.
Thank you.
Cheers.
All right, I will be sharing a slide. For all the participants, there is still a couple of minutes. We want to quickly introduce the organizations so that you can be a part of such exciting scientific research. Well, probably not on that level, but more on the undergraduate level. So I am going to quickly put up a slide, and I will hand over the mic to Adam. Give me 1 second.
Sure. Thank you so much for the introduction. For those who don't know me, I'm Adam Snyder, co-president of AMPC. The American Medical Physiology Club serves members providing resources they need to succeed, looking for research or internship opportunities. AMPC is GW's largest pre-health organization and has you covered. We partner with healthcare professionals to advise and guide members through weekly events that enhance our educational outcomes. As a pre-health community that aims to uplift one another, we offer career services to our members. These services include resume and CV to succeed. Sites more our training program is where our community thrives. Members are able to work and gain community service hours while also simultaneously making new friends over Zoom. Lastly, AMPC Symposium, our flagship research program, organizes students into teams to work on research projects with senior researchers overseeing their research. Each year, teams present findings to an audience of researchers, physicians, and students on GW campus and pursue publishing with GWU AMPC's assistance. If this is of interest to you, please join us on Engage and follow us on Instagram page for the latest updates and emails. I'll turn it back over to Divyat to give an introduction of GWiGEM.
All right, just give me 1 second. I'm going to pull something up right over here. Thank you so much, Adam, for that wonderful introduction. I will be talking a little bit about GWiGEM. Oops, sorry. Present real quick. I apologize, my internet is failing me, and okay, there it is. I think we're back again. All right, so I'm going to talk a little bit about GWiGEM. I am the president of GWiGEM, and we are a student-led organization aimed towards the revolution of genetic engineering to solve tomorrow's problems and to compete in the international genetically engineered machine competition. We're currently we're currently a team of 10 members and 3 PIs, along with 3 graduate student advisors. We started off in 2018 as a small club with only 5 members, and over the years we've kind of expanded and grown. We annually take part in this international genetically engineered competition, or also abbreviated as iGEM, part of our name. And this year we'll be taking part. This conference is taking place right now, so it's from the 14th of November to the 22nd. And this year, our project focuses on using K. michiganensis R19, which is a bacteria found in the Michigan area, and we're going to be using this to help filter lead ions and detect them in water, since lead water contamination is a big problem. Now, this bacteria has a natural tendency to aggregate lead ions to form a solid lead. And by transforming this bacteria, we hope to achieve a sensing capability and develop a device that can help filter and also detect lead. Over the past year, we've been working actively to develop this concept, which we will be presenting this Monday at the conference. And hopefully in the months to come, we'll be able to execute our plans and create this device. Anyone who's interested can can scan this QR code. Please follow us on Instagram, all our social media, and also look out for our emails. We should be starting to recruit for next year soon. So hopefully I am looking forward to see many of you in iGEM. Thank you.
If you have any questions about iGEM or AMPC, feel free to stay back and ask any of the members. But otherwise, thank you guys all for coming. I really hope you enjoyed it. And yeah, you're free to go if you don't have any questions.
Please come to our next event at 5 p.m. We have one with AMPC with Dr. Cantor, really cool physician from Kaiser Permanente Medical School.
Wow, AMPC is really like rolling out events on like that's amazing.
Thank you. Thank you for guys. I know, me too. Oh, it's Anshul.
Oh,
Hi, Anshul.
it's Anshul.
Yeah.
Who's anonymous?
How are you doing? He's one of our e-board members. That's good.
I like the fact that it says anonymous user. He joined midway through the talk and I kind of panicked. I was like, is our meeting
Me too.
I'm gonna stop recording.